I'm Sven, as Marcus said, and I want to just discuss the role of genetic diversity in biological control agents and the effects that genetic diversity can have on aspects of their biology and things. So, genetic diversity, control agents are sometimes found to have quite low levels of genetic diversity and can be a result of a number of factors, namely at the collection point in the native range, where they're often collected in small numbers and from a very few number of sites. Also, from population declines in mortality in transit, quarantine, mass rearing facilities, and also inbreeding from those same places. But we often refer to this as a genetic bottleneck. And the problem with that is that, well, the genetic bottleneck will decrease diversity in control agents. And this has a number of results. They can be beneficial or detrimental. In their beneficial forms, they can purge alleles from populations which may increase their efficacy. In the detrimental form, they can decrease the fitness of the control agent. And in introduced populations, this would increase their susceptibility to changes in the environment. So my study wanted to investigate genetic diversity and its effects on life history within a biological control agent. And in order to do this, we used Phenrica guerini, the biological control agent on Pereskia aculeata, which Ian Patterson spoke about yesterday. It's currently the only control agent released currently. The only agent released on Periskia. It's from South America. It's monophagous. It has 3 larval instars, and the females, it's a female sex biased ratio, and the females are larger, which it also takes a bit longer for them to develop from pupation to adult. We were very lucky in that there are 2 strains that we have. The original strain is the progeny of the original releases made in the early 1990s. They were collected in small numbers from Brazil. They spent quite a lengthy period of time in quarantine, and there were no subsequent releases made up until the next collection that was made for the mixed strain. It was a more recent collection from a variety of sites in Brazil, collected in quite large numbers, and this was crossbred with the original strain. So what we have is a strain with low levels of genetic diversity compared to the other strain, which has a bit more. And we identified 2 areas of life history that which we'd be able to compare, that being the duration of development and the fecundity of the 2 strains. And we placed a number of larvae on 2 plants, and we sampled each day by removing larvae from the plants, taking their weights, their length, and also the head capsule width from the larvae. From day 20 up until they entered pupation, we were able to get individual weights and track them into pupation as well as out when they're closed as adults. For fecundity, we gained the sex ratio at eclosion, and we paired virgin males and females together, and were able to get data on the number of egg batches produced per female, the number of eggs in each batch, as well as the number of larvae that hatched from them. And the results were actually quite astounding. So here, the mixed population is the one with higher genetic diversity, and it reaches the second instar, the third instar, and it enters pupation sooner than the original population, which has less genetic diversity. We actually alluded to protandrous eclosion earlier, and that is one reason that we can use to explain why the original population would reach eclosion sooner than the mixed population, as the mixed population actually has the female sex bias on them. All of these results also seem to be significant from one another. The weight of the 2 populations, the original and the mixed strains, entering pupation and at eclosion, is quite a good indication of fecundity and health within the population. So often we will take a pupal mass, but as Phenrica pupates in the soil, we couldn't do that without really killing the insect. So entering pupation and eclosing as adults, we were able to show that the mixed strain is heavier in both instances when compared to the original strain, and these were also deemed to be significant from each other. In fecundity, the results of our tests showed us that the mixed population with higher genetic diversity produces a lot more eggs. They produce more egg batches, more eggs per batch, as well as more larvae hatching from the eggs. And it was also deemed to be significant from one another. So in conclusion, we found that a more genetically diverse strain in Phenrica has produced a faster duration of development. They were also heavier at pupation and eclosion. They had a higher percentage of their eggs hatch, and they produced more females. And this is great for biological control because we sort of want these insects to expand once we release them into the environment. And all of these factors over here will influence that. So the recommendations that we can take from this study is that, yes, genetics has a place, and it does play a big part in the life history and the biology of the insect, and that especially in the case of Pereskia aculeata, we should use the strain with more genetic diversity because it performs better than the original releases. So we should use the mixed strain for Pereskia aculeata. Thank you very much.
Any questions?
Sven, do you think it could also influence host specificity?
It's a question that often arises. Will genetics affect the host specificity? But with monophagous insects, they've evolved with their plant for such a long time that those types of genes often don't, if there are mutations on those genes, it's probably deleterious to the population, so they wouldn't increase within the population at too much of a rapid rate, and they would probably die out, those individuals that had it. So I don't think that it would affect specificity directly, but I think there's more studies that would be able to highlight that point a bit more.
Especially since there weren't really any concerns about the specificity of Phenrica originally. Maybe that shows that it is really, they are all the same.
I'm sorry, I'm not quite...
The original strain that was tested for host specificity didn't have any non-target effects.
No, they weren't. It was deemed to be strictly host-specific in the original strain. And, we just, little side projects, we have given the mixed strain other plants, and they haven't eaten it at all. They've died straight away. So.
So, very interesting stuff. But one thing that I might have missed it, I was a wee bit confused, is the replication at the strain level, or so you were creating strains?
No, these strains were created. They were bred together by Sasri, by Denise and Des at Sasri, and we got individuals from them, from mixed strain, and our original strain was from one of the established sites at Port Alfred.
I'm just thinking, when you get to some conclusions someday, you might want to think about whether the replication should be at the strain level as opposed to within a strain. But because one of the things you might expect, if you had a vast number of low genetic diversity strains, there'd be much higher variability as well.
Yes.
Yeah, a lot of the agents might have even come from a single female. I think Trichanthium is possibly in this country, abundant just from a single female, and probably near Lepidopteris as well. And both of them as well. Eccritotarsus. So the trade-off also to increase genetic diversity is by bringing in more material with disease potential as well. So
yes, I mean one of the sort of the solutions to maybe increasing genetic diversity has often been to think about multiple introductions and reproductions, but there are some concerns there. As we've seen, genetics can affect the life history. So that's my sort of question.
So it would be sort of case by case, and make sure you've got your ducks in a row before you do it.
Right, thank you very much, Sven.